visit to the woods on a spring day is a great way to experience nature. Here we are surrounded by familiar forms of life, the trees and flowers of the plant kingdom, the birds, insects, and other moving organisms of the animal kingdom. But another entirely different kingdom of organisms also lives here, concealed by their small size and secretive habits. They are the kingdom fungi. A fallen log on the forest floor reveals the hidden world of fungi. They grow as thread-like structures called hyphae, and they are everywhere, in the soil, on the surface of most organisms, in every rotting creature. The thread-like hyphae of most fungi are microscopic in size. Here, time-lapse reveals their growth. Like plants and animals, fungi are eukaryotes. Their cells have nuclei. In some species, the tubular hyphae are long and have many nuclei inside one cell partition, a condition called cenocytic. Most fungi have partitions that break up the hyphae into smaller units, and still others are single-celled. Fungi are absorption feeders, releasing their digestive enzymes directly into the surrounding environment. The molecular products of digestion are absorbed back through the cell wall. Since these powerful digestive enzymes easily break down cellulose and lignin in the log, the cell walls of fungi must be made from a different material. It's a polysaccharide called chitin. Being absorption feeders, fungi must live in contact with their food, which is often decaying plants or animal material. But what about the ancient times, before there were any plants or animals? How and when did fungi evolve? Imagine traveling back in time some 600 million years. What would you find? In the oceans, a world swarming with single-cell organisms and simple colonies, but no plants and no animals. The largest masses of living material were clumps of algae. And by looking closely inside these clumps, you might discover an organism like this. Not much structure, just a few branching tubes. The cell walls are made of chitin, indicating these organisms are fungi. These ancient, simple organisms, neither algae nor plant, were the chytrids, the earliest of four main branches of kingdom fungi. Today they are found, like their ancient ancestors, in water. Here, chytrids digest the carcass of cyclops, a freshwater crustacean. In fact, chytrids play an important role in the decay and digestion of many aquatic animals. Recently, chytrids have been implicated in the serious decline of frog populations in many parts of the world. The chytrids grow inside cells of the skin and disrupt the frog's breathing. Chytrids are the only group of fungi with swimming spores or gametes. The flagellated cells are produced in ball-like sporangia, and when released, they swim off to find a new source of food. And here's another surprising fact discovered by sequencing their genes. Chytrids are more closely related to animals than to plants. On the tree of life, the evolution of plants, fungi, and animals looks like this. Another major split in the fungus line occurred about 500 million years ago. By this time, early animals were swimming in the seas, and plants were beginning to test life on the land. As animals and plants evolved, a new branch of fungi also evolved. A group that fed primarily upon the nutrients stored by these multicellular organisms, phylum Zygomycota. Today, zygomycetes are some of nature's most important decomposers, and they are particularly good at breaking down animal wastes. Feces, for example, is a rich source of nutrients because much of the energy stored in food passes right through an animal's digestive system. It's a good habitat for species of zygomycetes. The hyphae secrete digestive enzymes and absorb the products of digestion, turning much of this into spores. Hundreds of spores are produced in a spore chamber, or sporangium, elevated on a hyphal stalk. When the spores mature, being up in the air will help them be carried away by the wind to a new source of nutrients. You can see a real profusion of zygomycetes about a week after Halloween. The candle-cooked pumpkin is a treasure of nutrients. Hyphal threads penetrate and go to work. Elevated sporangia grow from every surface. Fruit flies, attracted by the fermenting pumpkin, inadvertently pick up the spores and transport them to other fruits in the neighborhood. It is virtually impossible to keep fruit from spoiling due to zygomycete spores, carried on the breeze or transported by flying insects. One rather amazing zygomycete fungus can be found out here in a cow pasture. Cows are somewhat fussy eaters, and they avoid eating grass that grows near a dung pile. It's a behavior that helps them avoid parasites. That's why you often see a ring of high grass surrounding each cow pie in a field. The zygomycete fungus in question is known as Pilobolus. It grows in moist cow plops, a perfect source of nutrients. The tubes are tipped with a fluid-filled bulb, and a black spore packet grows on top of that. But the Pilobolus living on any one cow pie have a problem: how to get their next generation into a new, fresh cow pie. The solution is to have their spores go through another cow, so that when the cow defecates, the spores are right there in the dung, ready to germinate. Now you can see the problem. Cows don't eat grass near a dung pile. So how can spores produced by Pilobolus on the dung pile end up in another cow? The solution? Fire the spores away from the cow pie onto fresh grass. And that is exactly the function of the bulb beneath the spore packet. It's a pressurized water cannon, and when it explodes, the spore packet goes flying, clearing the dung by as much as 2 meters. But since cow pies are often surrounded by dense grass, we guess that Pilobolus aimed its spore packets through holes in the grass shield. To test this hypothesis, we placed a dry cow pie in a cookie tin and added some water to activate the Pilobolus spores. We lined the lid with white paper, cut a central hole, and placed it on top. Then we put a bright light source over the hole. 2 days later, here are the results. No matter where it grew on the cow pie, Pilobolus aimed its spores toward the light source. It does this by bending the stalk, a behavior controlled by a light-sensitive region at the base of the bulb. When this region is completely shaded by the spore packet, the aim is correct. This behavior assures that spores are fired through gaps in the tall grass around a cow pie. In a fascinating way, this dispersal strategy serves more than just the fungus. Most cows have parasitic gut nematodes that send their eggs out in the cow feces. When the young nematodes hatch, they climb up the Pilobolus tube over the bulb and onto the spore packet, where they can now catch a free ride to their next host. But you don't have to hang out in cow pastures to find zygomycete fungi. You'll often find an exceedingly common one in your kitchen cupboard: Rhizopus stolonifer, the common bread mold. Rhizopus finds new bakery items by means of airborne spores. Once the baked good is invaded, it won't be long before black sporangia full of spores are formed. The hyphae are haploid, that is, their nuclei have a single set of chromosomes. These spores are produced by asexual reproduction. Chytrids and zygomycetes were the early branches of the kingdom fungi, but 2 more branches evolved around 300 million years ago: the ascomycetes and the basidiomycetes, mushrooms and their relatives. A close look at grapes in the early fall shows a dusting of white. actually a coating of ascomycete fungi. The microscope reveals yeasts, the important group of single-celled fungi that help us make bread, wine, and beer. Yeasts reproduce by asexual division. There's one dividing now. Another type of ascomycete can be found on rotting oranges. This species can also be grown in culture, and when this was done in the 1940s, it was discovered that this mold, Penicillium, produced chemicals that killed or suppressed competing molds and bacteria. This was the beginning of antibiotics. A different species of Penicillium is prized for the flavor it imparts to blue cheese. Most of the 30,000 or so species of ascomycetes are multicellular and have fruiting bodies where they reproduce sexually. These morels are edible, but similar false morels and elephant saddles can be deadly poisonous. Many ascomycete fruiting bodies are cup-shaped. If you cut a thin section through the cup, you can discover the characteristic structure of the group, the ascus, a sac containing ascospores. 4 daughter cells are produced by meiosis, and they divide again, giving 8 ascospores in one ascus. The advantage of a sexual life cycle is that it keeps reshuffling the genetic deck, allowing an ascomycete to be adapted for the particular demands of its environment. For example, this cup fungus grows on dung piles, and like Pilobolus, it shoots its spores away from the dung, where they are more likely to be consumed and passed into a new dung pile by another grazing animal. About half of the ascomycete fungi discovered so far are actually members of an intimate partnership. Instead of a saprophytic lifestyle, where they break down organic matter and slurp up the digested chemicals, these fungi rely on the food from photosynthesis. They're not photosynthetic themselves, but they are host to tiny green algae that do the work. Slice up a lichen, add a drop of water, and look for a layer of green algae cells just below the surface. Here, energy-rich molecules made by the algae during photosynthesis are shared with the fungus. This symbiotic relationship makes it possible for lichens to live on almost any landscape. Some lichens reproduce asexually by shedding little clumps of cells that contain both species. Landing on new territory, these clones will gradually grow into a new lichen. Lichen growth is so slow that some lichen patches are estimated to be several thousand years old. Lichens have specific mineral requirements. These bright orange lichens only grow where there is an abundance of phosphates, something there is plenty of in cow urine, as seen on the lower boards of this old milking barn. While most lichens are host to green algae, some, such as the lichens on these oak trees, contain cyanobacteria. The symbiotic cyanobacteria have the biochemical skills to do something that trees and algae cannot. They can convert nitrogen from the air into nitrogen compounds usable by other organisms. When they fall to the ground, lichens decompose, releasing their nitrogen into the forest. This slow but constant addition of nutrients is vital to forest ecosystems. The most familiar phylum in kingdom fungi is the Basidiomycota, the group that includes mushrooms. But some basidiomycetes look nothing like a mushroom. Corn smut is a parasitic fungus that takes over a ripening corn ear, turning the kernels into masses of spores. Rusts are a particular problem for wheat and other grains. This is a microscopic view of a wheat stem infected with Puccinia, wheat rust. Here is the same species on a different host plant, Berberis, showing Puccinia's complex life cycle in 2 different hosts. There are over 16,000 species of mushrooms, including some of the tastiest and some of the deadliest foods in nature. Naturally, one should never eat wild mushrooms unless they can be positively identified as an edible species. Eating this mushroom, Amanita muscaria, would make you very sick. This one, Amanita phalloides, could kill you. The name Death Angel is a fitting one. The mushroom that is visible is really just the fruiting body of the fungus, an elevated platform for launching airborne spores. Most of the fungus is underground, a mass of hyphae known as a mycelium. As the mycelium grows, activity is often concentrated at the outside edge, thus explaining the formation of a circle of mushrooms known as a fairy ring. The mycelia of some fungi are the largest and possibly the oldest living organisms on our planet. In one Washington state forest, a single individual honey mushroom mycelium was found to cover over 1,500 acres. Clumps of genetically identical mushrooms were found scattered over the entire area. The phylum name, Basidiomycota, comes from the microscopic spore-producing structures known as basidia, a word meaning club-shaped. A single mushroom may produce millions or even billions of spores. You can get some idea of the scale of spore production by making a spore print. The surface area for spore production is increased in several ways. Gills are a common method, with each gill bearing millions of basidia. Thousands of downward-pointing spines in this coral fungus also increase surface area. Pores can increase the surface for spore production. The boletes are one group of pore-bearing fungi. Another group is the polypores or bracket fungi. They're common in almost any forest, where they emerge from the bark of decaying trees. This one, chicken of the woods, is an edible species. Other basidiomycete fungi produce their spores internally, waiting for a disturbance to set their offspring free. One of the neatest spore dispersal tricks is found in the bird's nest fungus. It produces spore packets in a cup and lets raindrops toss them out. But the most ingenious spore dispersal is by fly trickery. Stinkhorns secrete a vile-smelling substance which holds great interest for flies. But the visitors can't avoid picking up some of the sticky, spore-laden ooze and carrying it to other locations. Stinkhorns come in a diversity of shapes, but they all have a foul smell that attracts flies. If you go searching for edible mushrooms, you'll soon find that some species are only found near certain kinds of trees. A king bolete at the foot of a pine, a chanterelle near a hemlock. If you look underground, you'll discover that the mycelium of the fungus is tightly pressed to the roots of the tree, or even threaded inside the roots. This relationship is called a mycorrhizal association, and it usually benefits both tree and fungus. The tree gets water, nutrients, and minerals extracted from the soil by its partner, and the fungus gets food produced by photosynthesis. Thousands of plant species have joined forces with mycorrhizal fungi. In fact, it's thought that plants could not have invaded the land without the mycorrhizal partnerships they formed with fungi in the harsh landscape of 400 million years ago. This plant, Indian pipe, has no chlorophyll, so it can't make its own food, but it can tap into the mycorrhizal association formed by a tree and a fungus, using the fungus as a conduit to steal food energy from the tree. Fungi evolved over 650 million years ago, becoming one of the three dominant kingdoms of multicellular life. They are eukaryotes, usually with a thread-like body that digests food externally and transports it inside through cell walls made of chitin. Fungi are key ecological participants on planet Earth. They recycle dead plants, they form partnerships with algae to pioneer the landscape, and they join forces with trees to form powerful mycorrhizal associations. Fungi are remarkably successful, with over 70,000 known species and perhaps 1 million yet to be described. With 4 main branches: chytrids, an ancestral aquatic group; zygomycetes, a branch that includes many of the molds; ascomycetes, the cup fungi and true yeast; and basidiomycetes, the mushroom-forming group. The fungi provide a fascinating, diverse, and often mysterious kingdom of life.